Hi, hello students, how are you? Welcome to MNR Physics. This is your MRK Zoology faculty. So now today we will continue the part 2. So in previous class we have discussed about part 1 in human male reproductive system. Now we will continue to the part 2, the continuation of human male reproductive system. In previous class, we have discussed about what is then up to testes. See, the human male reproductive system constitute with one pair of testes and accessory ducts, and what are next these accessory glands and urethra and penis. These structures are there in the human male reproductive system. In previous class, we are already started and already discussed about the testes and internal structure. Once again, I will show the brief explanation regarding these these male reproductive system character, and then along with this, we will continue the remaining characters. Okay. So next continuation here. See, in previous class we are discussed about, yes, we are continue about these testes internal structure, and in previous class we are discussed about up to the what is accessory ducts. See, the accessory ducts are start from what is seminiferous tubule, and seminiferous tubules to what is seminiferous tubules are the structural and functional units of male reproductive system. So what is the reason for the seminiferous tubules are called as a structural and functional unit of male reproductive system? Means within the seminiferous tubule, what are the spermatogenesis process will initiate, or the sperm production is takes place within the seminiferous tubule. And we are clearly discussed in previous class the transfer sectioning of testicular lobule, and inside the seminiferous tubule how many types of cells are there, outside the seminiferous tubule how many type, what type of cells are there, we are clearly discussed. Okay, and inside the seminiferous tubule we are discussed exactly two types of cells are there. One type of cells are the Sertoli cells, and the second type of cells are the what are spermatogonial mother cells. Okay, and outside seminiferous tubule Leydig cells. And what are the role of these testicular lobule, what is Sertoli cells and spermatogonial mother cells? And already we have discussed. Once again, I'll give the brief information regarding Sertoli cells as well as spermatogonial mother cells and outside seminiferous tubule leading cells. See, first Sertoli cells. Sertoli. See, these Sertoli cells are present inside seminiferous tubule, and these are helps to nourishment. These are involved in the nourishment. See, nourishment means what? These Sertoli cell secretions are contain some kind of nutrients. That nutrients are helps to growing spermatozoas. The spermatozoas are matured inside the Sertoli cell. Means the total differentiation process will completed within the Sertoli cell. Nourishment cells. Okay. Next, spermatogonial mother cells. Spermatogonial mother cells are there. These spermatogonial mother cells, which are continuous to the spermatogenesis process. Spermatogenesis. Spermatogenesis means the sperm production is takes place here. The sperm production is takes place from the spermatogonial mother cells, and along with this, and these two types of cells are present inside seminiferous tubule. Next, Leydig cells are there. These Leydig cells are present outside seminiferous tubule, and these Leydig cells are also called as a interstitial cells. These are also called as a interstitial. These are also called as a interstitial cells. What is the function of these interstitial cells in these means? These are involved in the production of androgen. That androgen is the testosterone. Testosterone hormone production takes place by the Leydig cells. So these are the important points regarding inside seminiferous tubule. These two are inside seminiferous tubule and it is present outside seminiferous tubule. So these are the important points regarding what is the seminiferous tubule. And after completion of the sperm production, okay, we will discuss in the spermatogenesis and the detailed information. Next, as per this picture, after completion of the spermatogenesis process inside the seminiferous tubule, the sperms are enter into the from seminiferous tubule to the sperms are enter into the remaining accessory ducts. So what are the remaining accessory ducts? Already we are clearly discussed that. See, from seminiferous tubule to the sperms are enter into the rete testis. See, the process of movement of spermatozoa from seminiferous tubule to into the rete testis, that process is called spermiation. Spermiation. Especially based on this spermiation, spermiogenesis, spermatogenesis, erection and ejaculation. Based on these five words in our NEET exam, 100% that questions are there. Especially in 2021 NEET exam, one question is there on this spermiation and spermiogenesis. What are the questions on these what is spermiation and spermiogenesis? Based on true or false statement given that questions. True or false statement. So spermiation, the statement 1, spermiation, the process of movement of sperms from seminiferous tubule to into the next accessory ducts, that is right. And statement 2, spermiogenesis, okay, the process of conversion of spermatids into spermatozoa, that is called a spermiogenesis. Based on these two words, question is there in 2021, question is there in 2021 also, one question is there on these words. I'll explain detailed information this. And order of chronological order of movement of spermatozoa from inside testis to outside. Okay, from seminiferous tubule to it is these sperms are enter into the rete testis. Rete testis 2, again the sperms are enter into the vasa efferentias. Vasa efferentia 2, the sperms are enter into the what is epididymis, and the epididymis structurally here divide into three parts here, right? Which part of the epididymis, okay, which part of the epididymis which is taking the spermatozoas, which part of the right epididymis which is taking the spermatozoas from inside testis to outside, okay, that is the capsule epididymis, that is the capsule epididymis, that is one. And the next it is the proximal portion of the epididymis. Generally the epididymis is the highly coiled portion, it is highly coiled portion, it is one. The proximal portion of the epididymis and that portion which is connected with vasa efferentia, that is capsule epididymis. The capsule epididymis which is collecting the spermatozoa from inside testis to outside through the vasa efferentia. Some questions are there, I will frame after the explanation. And after capsule epididymis, next the capsule epididymis is open into the middle long coiled portion of the epididymis, that is the corpus epididymis. Now the corpus epididymis is also highly coiled structure and which is again finally open into the end part or distal part of the epididymis, that is the chorda epididymis. Chorda means that is the tail portion, okay. So finally, the chorda epididymis is open into the uncoiled portion of the spermatic cord. The uncoiled portion of the spermatic cord is called the vasa differences or vas differences. It is the vas differences. It is vas deferens. It is uncoiled portion. This epidermis is the coiled portion, and these all structures are constitute called as a, that is what, accessory ducts. Means capsule epidermis to carpus, carpus epididymis to cauda, cauda epidermis to vasa deferences. These vasa deferences finally it is open into the vasa deferences. It is open into the ejaculatory duct. It is open into the ejaculatory duct. That is open into the ejaculatory duct. Okay. So the ejaculatory duct that is open into the urethra. I'll show in the next picture, that is. Okay. So these are the, then what is the next ejaculatory duct here? Right? Yes. This is the ejaculatory duct. It is the ejaculatory. It is the ejaculatory duct. These it is the vasa differences. It is the vasa differences. It is vas differences. These vas differences it is open into the ejaculatory duct. The ejaculatory duct which is again open into the urethra. Urethra is open out through the urethral meatus. Okay. So these are the important points regarding this. It is very clear. Okay. And this is the structural information about the testis. It is the structural information. See, actually, what is the up to epididymis? These are all are present inside the scrotum. Okay, within the lower abdomen, within the scrotum. From scrotum to into the lower abdomen, into the abdomen, the vasa differences is extend. Vasa differences structure is ascend into the abdomen from the scrotum. Okay, through which structure the vasa differences it is ascend into the abdomen. Means here one structure is used for the ascend the spermatic cord. The ascend that structure is the inguinal canal. The inguinal canal it is act as a bridge. It is act as a bridge. It is act as a bridge. See, if the inguinal canal absent, from scrotal sacs to into the abdomen, the spermatic cord cannot be ascend. Ascend means it is not move up from bottom scrotum to into the abdomen. Okay, so it is very clear. It is inguinal canal helps to movement of this spermatic cord from in what is this, scrotum to into the what is abdomen or lower abdominal cavity. So this is the important thing. So it is one clear. Then what is the chronological order of, what is the chronological order of movement of spermatozoa? The sperm movement begins from which accessory duct and which is ends in which accessory duct? That is the question. That is the chronological order. The sperm production takes place within the seminiferous tubule. That is clear. Okay. After that, the seminiferous tubule to that is enter into the rete testis. Rete testis to that sperm is enter into the vasa afferentia. Vasa afferentia to that is enter into the epididymis. Epididymis finally it is open into the what is vasa differences. This is okay. Vasa differences that is open into the ejaculatory duct. That is open into the urethra. Urethra to that is released into the female reproductive tract by the mating or copulation process. So this is the order of movement of spermatozoa from a seminiferous tubule to into the next accessory duct. Okay, it is one. After that, so here we are used one word. What is that word? Is the spermatic cord. Spermatic cord. It is made by. It is constituted with which structure? Okay, spermatic cord constitute with which structure? See, the spermatic cord it is. Okay, the spermatic cord constitute with the vasa differences, cremaster muscles, nerve cord, blood vessels, and lymphatic cord. These structures are present in the spermatic cord. These structures are present in the inguinal canal, and through the inguinal canal some structures are enter into the scrotal sacs from the abdomen, and some structures are enter into the abdomen from the scrotal sac. Okay, and in these spermatic cord structures, which structures are enter into the scrotal sac from the abdomen? That is question. And from abdomen to scrotal sac, what are cremaster muscles, nerve cord, blood vessels? These three structures are enter into the scrotal sac from the abdomen. And from scrotal sac to abdomen, which structures are ascend? Vasa differences and the lymphatic cord. See, the lymphatic cord is origin within the where the lymphatic cord is origin. The lymphatic cord is origin within the what is scrotal sac, as well as the vasa difference also origin within the scrotal sac. That's why from the scrotal sacs to into the abdomen, vasa differences and lymphatic cord, and from abdomen to into the scrotal sac, cremaster muscles, nerve cord and blood vessels. What is the importance of cremaster muscles? So these are having main role in the maintenance of temperature within the scrotal sacs. Generally, the cremaster muscle function is depend on the environment temperature. For example, when our surrounding the body temperature is present, our surrounding environment temperature is lesser than our body temperature, these cremaster muscles are become construct and the scrotal sac diameter becomes reduced, thereby the normal temperature is maintained surrounding the testes. And compare with our body temperature within the environment, if high temperature is present, these cremaster muscles are perform the relaxation, thereby surrounding the testis large space is developed, thereby less temperature is appeared in the surrounding the testis within the scrotum. The less temperature is helps to production of spermatozoa, means initiation of spermatogenesis. Okay, so these are the important points regarding these what is spermatic cord. This is one, and after this next we'll discuss what is this structure of what is male reproductive system. Here it is complete entire male reproductive system structure it is. Okay, the total male reproductive system constitute with one pair of testis, accessory ducts. Okay, we are discussed about these points here, and the next we will discuss the continuation of accessory ducts, what is accessory glands and urethra, penis. Now we will discuss this structure. Okay, come to the point. This is the vasa differences. These vasa differences it is ascend into the abdomen. Actually, the vasa differences it is overlap to the what is urinary bladder, and it is enter into the these vasa differences open into the ejaculatory duct. Okay, now where the ejaculatory duct is there, here little swollen structure is there. It is also seminal vesicle. It is the swollen structure. Okay, it is the ejaculatory duct. It is the swollen duct, swollen portion. So, what is the reason the ejaculatory duct is present in swollen in nature? Okay, one reason is there. So, the seminal vesicles are open into the ejaculatory duct. Okay, the seminal vesicles are can secrete the seminal fluid. The seminal fluid contain the some nutrient. Okay, that nutrients are helps to what is maturation of spermatozoa. Okay, the sperm cell can take the nourishment. Temporarily the sperm cell can store in this what is the swollen structure, up to which time the sperm cell can store here, and that much time the sperm cell can receiving the nourishment, and that nourishment is present in the secretion of seminal vesicles. That is clear. That's why the ejaculatory duct is present in swollen structure. Temporarily the sperm cell can store in ejaculatory duct. Okay, so this is one point. After that, okay, the seminar, then the ejaculatory duct is finally both ejaculatory ducts are which are open into the urethra structure. Okay, so and first we will discuss the accessory ducts are male accessory gland. Accessory glands are male reproductive glands. accessory glands. Accessory glands are male reproductive glands. Male reproductive. Male reproductive glands. Male reproductive glands. So total how many are there means three types. Three types of male accessory glands are there. In these three types, okay, one type of accessory glands are the seminal vesicles. Seminal vesicles. These are the seminal vesicles. And the second type of accessory glands are prostate gland. It is only one. Prostate gland. And the third type is the bulbourethral gland. Bulbourethral. Bulbourethral gland. These are in these three types of male reproductive glands. Okay, seminal vesicles are the paired. Here, how many are the paired? How many are the unpaired? So these are one pair are there, and prostate gland is the single gland, and bulbourethral gland is also what is paired. See, in these total types of glands, how many are the paired? How many are the unpaired? In these three types of male reproductive glands, two types of glands are the paired. These are seminal vesicle and bulbourethral glands are the paired. And the prostate gland, it is the unpaired gland. So it is also called as called with one more name, that is Cowper's gland. That is the Cowper's gland. This is the unpaired gland. I'll show in this picture these three types of male reproductive glands. See, it is one seminal vesicle, one pair are there, and bulbourethral glands again here one pair are there. These are one pair seminal vesicles either side of the urinary bladder, and either side of the urethra again bulbourethral glands are there. Okay. Next, unpaired Cowper gland. This is the prostate gland. See, it is present posterior to the urinary bladder. Posterior to the urinary bladder, the prostate gland is there. Total three types of glands are there. In number five are there. In number five types, three types. Okay, in number five types, three types here. Means number means two seminal vesicles and one prostate gland and two bulbourethral glands. Two, one, two. Total five in number. And types how many? Total three types are there. And in these three types of male reproductive glands, how many are the paired? How many are the unpaired? Unpaired male reproductive glands are only one type. These are the prostate or Cowper gland. And paired reproductive glands are only seminal vesicles and bulbourethral glands. Okay. These three pairs of these three types of male reproductive glands can produce 100 percentage of seminal fluid. Okay, 100 percentage of seminal fluid is produced by these three types of male reproductive glands. Okay, so what is the chemical composition of seminal fluid? Chemical composition. Chemical composition of what is accessory glands or seminal fluid. Chemical composition of seminal fluid. See, it is a alkaline, slightly alkaline. Okay, it is a slightly alkaline. It is a slightly alkaline nature. It is one slightly alkaline. And within the seminal vesicle, the major component is the fructose. Fructose is the major component. Along with this fructose, calcium ions are also there. Along with the calcium ions, certain enzymes are present. Along with the components, certain enzymes. Okay, certain enzymes are there. So this is the chemical composition of seminal fluid. See, these 100 percentage of seminal fluid constitute with this structure. So slightly alkaline. Okay, slightly alkaline means what here? So 6.6 to 7 odd is between that, means 7 point approximately 6.8 to 7.2 exactly, that is. That's why we are not considered slightly acidic, and we are not considered slightly exactly, we are not considered acidic, which exactly we are not considered acidic. That's why sometimes we are called as a slightly acidic, and sometimes we are called as a slightly acidic and slightly alkaline. Two words we are used here. Okay. So right, and what is the color of the semen? The semen color is, okay. The semen color is, what is seminal fluid color? It is seminal fluid color. Color of seminal fluid, that is whitish in color. Okay, color of seminal fluid, white color. White color it is. Color of seminal fluid, white color. So it is. So what is the reason, the presence of which male reproductive glands are giving the whitish color? That is the question. Say the prostate gland secretions are giving the whitish color. Prostate gland. Prostate gland secretions. Prostate gland color secretions giving the whitish color, and seminal fluid color along with this. Okay, so the seminal fluid color is whitish color, then the presence of which male reproductive gland secretions, the seminal fluid color is in whitish in color, means the prostate gland secretions are giving the whitishness color or whitish color to the seminal or seminal fluid. It is one. And here one more important point is there. What is, in the total 100 percentage of, in the total 100 percentage of what is the seminal fluid, in the 100 percentage of seminal fluid, okay, and almost 60 percentage of seminal fluid from the seminal vesicles, seminal seminal vesicles, okay, and remaining 15 to 30 percentage of seminal fluid is from the, what is bulbourethral, bulbourethral and prostate gland, bulbourethral and prostate gland. It is one. And what is the high amount of, what is fructose is there in the seminal fluid? The fructose concentration is there, the fructose production is from the seminal vesicles secretions, seminal vesicles. And what is the color of seminal vesicular fluid? Means waterish color, colorless, that is. And bulbourethral gland secretion also waterish color. There is no whitish in color. Only the prostate gland secretions are giving the whitishness to the seminal fluid. That's enough. Right. Now, one more important point is there. What is semen? Okay, semen means seminal fluid plus spermatozoa. Seminal fluid, seminal fluid plus spermatozoa. Seminal fluid plus spermatozoa constitute called as a semen. Okay, so this is the important thing. Semen is contains seminal fluid and spermatozoa. These are present within the semen. The semen can be ejaculate into the female reproductive tract during the copulation. Okay, so this is the seminal fluid detailed information. Generally, in our examination asking regarding the semen, what is the composition of seminal fluid? What is the semen? And What is the highest contribution? Which male reproductive gland has highest contribution for production of seminal fluid? The highest contribution is by the seminal vesicles. The next contribution by the bulbourethral glands as well as prostate gland. It is one. And one more important thing, say the bulbourethral glands have a secondary function. Okay, the bulbourethral glands involved in two functions here. One function for the seminal fluid, it is seminal fluid contribution, and also the bulbourethral glands helps to these gland secretions helps to lubrication lubrication and it is act as a flushing agent within the urethra. Flushing agent. Flushing agent. See, flushing agent means what? See, it is the bulbourethral. These are the it is the bulbourethral gland. Okay, it is the bulbourethral gland. The bulbourethral gland secretions are there. These secretions are what is contribution? The pulvital gland contribution, two contributions are there. One contribution for the seminal fluid formation, okay, seminal fluid formation. Second one, lubrication and flushing of the urethra. See, after maturation, some urine particles are can store inside the urethra. See what happens if the urine particles are cannot removed. Okay, see, if the urine particles long time can stored inside the urethra, the inner lining of the urethra can be damaged by the urine. So urine means urea is there. The urea can damaging the tissue cells. That's why after maturation, the bulbourethral glands are can release some secretions. That secretions are helps to flushing out the remaining urine particles from the urethra. Remaining urine particles from the urethra. So in this way, the urethra will be protected by the secretion of bulbourethral glands. Okay, so that's why the bulbourethral glands have two functions. One function contribution in the seminal fluid. Second function what is protecting the inner lining of the urethra. Protection of inner lining of the urethra. So these two functions are done by bulbourethral glands. It is one point here. Okay, and so these are the some important points regarding seminal fluid. And after that, next we will discuss. It is the male reproductive glands. And after that, next go to urethra. Okay, next go to urethra. See, urethra is there. Okay, urethra. Generally, in male reproductive system, the urethra is in long. Long urethra is present in male reproductive system. Okay, so I'll write these points here. In male, long urethra is present. Long urethra. In male, long urethra is present here, approximately 20 centimeters long. Approximately 20 centimeters long it is. Long urethra is present, it is one. And I'll compare with the female urethra. In female, short urethra is present. Urethra is short. Short urethra is present. Approximately it is present 4 centimeters. Very short urethra is present. What is the reason the long urethra is present here? Means it is extend into the penis. It is extend into the penis. That's why the male within the male reproductive system, the urethra is in very long, approximately 20 centimeters. Okay, and this long urethra can be divided into four parts here. This long urethra can be divided into four parts. Okay, what are the four parts in this? Okay, in this one is the urinary urethra. One is the urinary urethra, and second one is the prosthetic urethra. Prosthetic urethra, and third one is the membranous urethra. Membranous urethra, and fourth one is the penile urethra. So these four parts are there: urinary urethra, prosthetic urethra, membranous urethra, penile urethra. I'll draw the one picture for easy understanding this. For example, it is the urinary bladder. It is, and the urinary bladder, it is open into the urethra, and into this urethra again, which are opened by the two ejaculatory duct from either side, again which is continue into the penis. Again it is continue into the penis. It is one. Okay, it is one point. And here, what is in this region? Here the prostate gland is present, and at which region bulbourethral glands are there? Bulbourethral glands are there. It is one. Next, this portion is the penis. Okay, I'll explain this. Right. See, at which part of the urethra it is open from the urinary bladder? It is the first one, that is urinary urethra. This is the urinary urethra. And next, which part is open into the urethra of this prostate gland? That is the prosthetic urethra. Generally, it is the prostate gland. The prostate gland open into the urethra with the help of prosthetic duct. This prosthetic duct is open into the urethra. So, this portion is called the prosthetic urethra. After that, what is, where does the ejaculatory duct and bulbourethral glands are open into the urethra? This is the membranous urethra, and remaining large portion of the urethra is the penile urethra. The penile urethra is very long. Penile urethra is very long in this. That's why in male reproductive system, long urethra is there. Okay, and based on urethra, the male reproductive system is also called with one more name. That name is urinogenital system, and this duct is the urinogenital duct. This duct is also called as a urinogenital duct. Okay, what is the reason this duct is called urinogenital duct? Means through the single urethra, it is receiving the sperms from the ejaculatory duct and also transport the urine from the urinary bladder. Say this is the urinary bladder. It is the urinary bladder. Okay, so the urethra has two functions. It is urethra can performing two functions here. One function is passing of urine, and second function is transportation of semen into the female reproductive tract. Through the single way, two functions are done. That's why this urethra is called as a urinogenital duct, and the entire male reproductive system is called as a urinogenital system. So this is the urethra structure. Four parts of urethras are there: urinary urethra, prosthetic urethra, membranous urethra, penile urethra. In these four parts of the urethra, long portion of the urethra is present in the penis. Long part of the urethra is present in the penis region. So this is the, what is, urethra structure. This is the urethra, and after that we'll discuss next another male reproductive system, male reproductive organ, that is the penis. This entire structure is called penis. This entire structure is the penis. Okay, this entire structure. Generally, the penis, it is made by mainly two types of tissues are there here. Two types of tissues are involved in the formation of these what is penis here. See, this is the urethra. As usually it is the urethra. See, surrounding the urethra, corpus spongiosum tissue is present, and below to this urethra, corpora cavernosa connective tissue is present. Okay, so corpus spongiosum, corpora cavernosa, these two types of tissues are present inside the present surrounding the urethra. That is one point here. Actually, these corpora cavernosa and corpus spongiosum, these both are highly vascularized. These both are highly vascular portion. Highly vascularized means what? More blood vessels are there. Blood capillaries are more. Blood capillaries. Okay, blood capillaries are more. See, what is advantage the presence of blood capillaries means during the copulation by the heavy rush of blood into the corpora, what is it, cavernosa and corpus spongiosum, the penis becoming a rigid. The process of rigid of penis by the heavy rush of blood into this tissue, that is called erection. That erection is helps to rigid of penis. The rigid penis is helps to insemination into the female reproductive tract. That's why the penis is also called as a introminant organ. Means it is helps to transfer of semen into the female reproductive tract. It is helps to transfer of semen into the female reproductive tract during the copulation. So this is the penis structure. It is a penis. Okay, and finally the tip of the penis, it has a, it is a one pinkish color structure is there. The pinkish portion of the penis is called the glans penis. It is the pinkish portion. It is the glans penis. Okay, and the total penis and glans penis which are covered by the one thin skin, the skin structure is called here foreskin. The total penis and this pinkish color structure is covered by the skin. The skin is called a foreskin. It is one. And along with this, the opening of urethra, okay, that is called urethral meatus. That is the urethral meatus. It is the opening. Through this urethral meatus, the urine comes out from the urethral meatus, as well as semen also comes out from the urethral meatus. Okay, urine and semen, these both are comes out from the what is urethral meatus. That is the opening of what is opening outside opening, that is the urethral meatus. Okay, so these structures are present in the what is male reproductive system. Up to here, this is the structural information. Up to here, it is what structural information. Next we'll discuss the new points here. Okay, next we will discuss the new points. That new points are, first we will discuss about 5 points here, very important points these are. Okay, I'll explain to you. Right, first word is the, what is the meaning for spermatogenesis? What is the meaning for spermatogenesis? What is the meaning for spermiogenesis? Spermiogenesis. And what is the meaning for spermiation? And what is the meaning for erection? What is the meaning for ejaculation? Okay, these are there. Okay, we'll discuss these 5 points in 2020, 2021 and 2022 also. Okay, from these 5 points, every year one question is there in these last 3 years. Okay, see, what is the meaning for spermatogenesis? Okay, spermatogenesis means the process of conversion of the entire process of sperm production. Okay, process of conversion, process of conversion of spermatogonial mother cells, spermatogonial mother cells into spermatozoas. Spermatogonial mother cells are converted into spermatozoa. The entire process is the spermatogenesis. That is clear. Next, what is the meaning for spermiogenesis? Spermiogenesis means the process of, okay, conversion of, the process of conversion of spermatids into spermatozoa. So this is the what is spermiogenesis, and the total spermiogenesis process takes place within the Sertoli cells. It is completely takes place within the Sertoli cells. Okay. Next, what is the meaning for spermiation? Spermiation means the process of movement. Okay, the process of, okay, movement, the process of movement of sperm, sperms. Movements nothing but what is it is start the locomotion. Okay, it is start the locomotion, or it is also we are use that another word, the process when it is start the migration, migration of sperms from seminiferous tubules to next accessory ducts. Seminiferous tubules, seminiferous tubules to, okay, into next accessory ducts. Seminiferous tubules to into next accessory ducts. Next accessory, next accessory ducts. So that is the spermiation. See, after completion of the development of sperm inside the Sertoli cell, the mature sperm can detach from the Sertoli cell and which is start the migration, which is start the movement. Okay, so that is called spermiation. Next, go to erection. It is the process of rigid of penis by the heavy rush of blood. The process of rigid, the process of rigid of penis, the process of rigid of penis by heavy rush of blood, by heavy rush of blood, by heavy rush of blood into penis. Okay, it is the erection, the process of rigid, the process of rigid. Next, ejaculation. Okay, ejaculation means the process of releasing the semen, the process of releasing the semen into female reproductive tract, female reproductive, female reproductive tract. So that is nothing but vagina, female reproductive tract, nothing but it is the vagina. Okay, these five words meanings are this. Very important words. Very important meanings these are. Very important. So spermatogenesis, spermiogenesis, spermiation, erection, and ejaculation. These points are there. Okay, and after that we will continue the complete entire female reproductive, male reproductive system, that is Next, up to here only we are discussed about the structural information. Next, we will discuss the function, that is spermatogenesis. Next, we will discuss the spermatogenesis process. Next, we will discuss the spermatogenesis. Okay. So the spermatogenesis process, it is completed within, okay, spermato spermatogenesis. Okay, already we are discussed. Spermatogenesis means what? The process of conversion of spermatogonial mother cells into spermatogonia, that is called spermatogenesis. The entire spermatogenesis process completed within the seminiferous tubules. Okay, right. See, the spermatogenesis process, it is nothing but kind of gametogenesis. Now we are discuss the gametogenesis. Gametogenesis. Okay, the gametogenesis means the process of production of gametes is called gametogenesis. Okay, so production of male gamete, male gametogenesis, production of female gamete, female gametogenesis. The production of male gamete, what is the male gamete? Sperm. What is the female gamete? Ova. So the process of production of sperm is called spermatogenesis. The process of production of ova is called oogenesis. Now we will discuss the male reproductive system process, that is sperm production, that is the spermatogenesis process. Okay, it is also called as a, in this process it is also called with spermiogenesis or spermatogenesis is over. This point is over, and it is. Say, generally in human male, the spermatogenesis process begins at the puberty age. Okay, so very important points are there. Gametogenesis in male, it is started at the puberty age. Gametogenesis in female, it is started at the embryonic stage in female fetus. Female, when the female is present in fetal stage, gametogenesis is begin. But in male, at the puberty age only, the spermatogenesis process begin. Okay, I'll write the same words here. In males, okay, in males, spermatogenesis spermatogenesis begins at puberty age. Puberty age. Puberty age. What is the puberty age in human beings? 12 to 14 years age is the puberty age. 12 to 14 years age is the puberty age. And at this age, from the hypothalamus, okay, now I'll explain the hormonal regulation. Okay, hypothalamus is there. Okay, it is right. Hypothalamus. Hypothalamus is there. Okay, the hypothalamus at the puberty age only. At the puberty age, hypothalamus can release the gonadotropin releasing hormone. This gonadotropin releasing hormone, it is reached to the anterior pituitary. Anterior pituitary. Through which structure? What is portal circulation here? Okay, hypophyseal portal circulation is there through that. This anterior pituitary also called as a adenohypophysis. It is the adenohypophysis. Now this anterior pituitary can release, okay, gonadotropins here. It is release gonadotropins. Okay, so the gonadotropins are, one gonadotropin is the One gonadotrophin is the FSH, what is FSH, and second gonadotrophin is the LH. Now, follicular stimulating hormone and luteinizing hormone. And the luteinizing hormone is also called as a interstitial cell stimulating hormone. Okay, reason these luteinizing hormone act on Leydig cell. It is act on Leydig cells. Okay, so the Leydig cells also called as a interstitial cells. Interstitial. These are also called as a interstitial cells. Okay, the interstitial cells, that's why interstitial cell stimulating hormone, and the Leydig cells are involved in the production of, that is the male androgen. The androgen is the testosterone. Okay, androgen is produced, that is the testosterone. The androgen is the testosterone. Next, follicular stimulating hormone. What is the role of this follicular stimulating hormone? Which is initiate the spermato, spermatogenesis, and which is stimulating to the Sertoli cells. Sertoli cells. The Sertoli cells function is what? Nourishing. Nourishing. These are the nursing cells. These are the nourishing cells. Okay, Sertoli cells, these are the nourishing cells. These nourishing cells means nutrients. These nutrients are helps to maturation of spermatozoa. So, this is the hormonal regulation of spermatozoa. It is hormonal regulation. Hormonal regulation of spermatogenesis. Spermato Hormonal regulation of spermatogenesis. That is one. Right, up to here it is clear. Okay, okay. So at the puberty age, when the hypothalamus can release the gonadotrophic releasing hormone, the anterior pituitary can stimulated by this hormone, and which is released these two types of gonadotrophins, FSH and LH. See, the FSH hormone is stimulating to the Sertoli cells for the nourishing and also initiation of spermatogenesis, and LH hormone is act on the Leydig cells. The Leydig cells are involved in the production of androgen. The androgen hormone is the testosterone. The testosterone is again helps to spermatogenesis and maintenance of male reproductive characters. This is okay, and come to the point here. See, at the puberty age, the spermatogenesis process begin, and the spermatogenesis process completed by four phases here. In human male, the spermatogenesis process completed by four phases here. Four phases are involved in this. In these four phases, one is the multiplication phase. Multiplication, it is one, it is the multiplication phase, and the second one is the growth phase, third one is the maturation phase, and fourth one is the differentiation phase. Differentiation phase, it is also called with one more name, that is spermiogenesis. That is spermiogenesis. Spermiogenesis, it is the spermiogenesis. Okay, so in this, and say the multiplication process by mitotic division, here only mitotic division. Okay, mitotic division is there. Okay, and growth phase, there is no division. There is no division. And maturation phase, meiosis process. Meiosis. Meiosis process is there here. Meiosis. And differentiation, again, no division here. No division. Okay, no division. No division. Mitosis only in multiplication. In this process, what happened? The number of spermatogonial cells increase and the size of the cells increase, and the after completion of the growth phase, these are initiate the meiosis process. Okay, that meiosis, after meiosis, that spermatids are formed and the spermatids are converted into spermatozoa, that is the differentiation phase. Okay, now we will discuss the entire complete process of the spermatogenesis with the help of these four phases here. Okay, right. In this, first multiplication, it is the first multiplication phase. Multiplication. Multiplication. In this the multiplication, it is begins within the, then already we have discussed the seminiferous tubules are lined by two types of cells. One is the spermatogonial mother cell, second one is the Sertoli cell. The spermatogonial mother cells are initiate the what is spermatogenesis process. Okay, see, it is the spermatogonial mother cell, spermato, spermatogonial mother cells. These spermatogonial mother cells are present in diploid condition. Diploid condition means which are with what is 46 number of chromosomes are present in this. Next, these cells are involved in the repeated mitotic divisions. Repeated mitotic divisions, thereby the spermatogonial cells are formed again and again by the repeated mitotic, by the repeated mitotic divisions, again and again, again and again, these what is cells are, the spermatogonial cells are involved in the repeated mitotic divisions, and millions of spermatogonial cells are developed here. Millions of spermatogonial, millions of spermatogonial cells. Okay, these spermatogonial cells are also present in a diploid state, means 46 chromosomes are there. By the repeated mitotic division, okay, millions of spermatogonial cells are developed inside the seminiferous tubule. From here to here, this is the multiplication phase. Okay, see, in this multiplication by the repeated mitotic divisions, okay, so the spermatogonial sizes are minimized, means size is decreased. See, observe, the spermatogonial mother cell size is this size and spermatogonial daughter cell size is this size, means this size is decreased. So for increase the size of the what is the spermatogonial cells, these all millions and billions of spermatogonial cells are enter into the second phase. The second phase is the growth phase. Okay, in this growth phase, what happened? The small sized spermatogonial cells are what is develop, what is increase the size of the small sized spermatogonial cells. Okay, say after completion of the growth phase in this all, for example here 2 billions of or 2 millions of spermatogonial cells are there. 2 millions of spermatogonial cells are there. The 2 millions of spermatogonial cells entire at a time, these are completes the growth phase, and the size of the spermatogonial cell is increased. See, after completion of the growth phase, these cells are called primary spermatocytes. These are the primary spermatocytes. These are the primary spermatocytes. It is one. See how the size of the cell is increased in the after completion of the growth phase. That is one important point. See, actually in the spermatogonial cells, the chromatin and chromosome size and concentration is less. When these all cells are enter into the growth phase, the chromatin concentration is increased. Chromatin concentration, chromatin concentration increased. Chromatin concentration increased. By the increasing the chromatin concentration, nucleus size increased. Nucleus size. By the increasing the nucleus size, cell size also increase. Cell size increase. In this process, the cell size is increased in the growth phase. After completion of the growth phase, that all spermatogonial cells are developed or converted into what is primary spermatocyte. Means here 2 millions of spermatogonial cells are enter into the growth phase. Here again 2 millions of primary spermatocytes are produced, because in this growth phase there is no cell division process. There is no cell division. Okay, how many number of spermatogonial cells are enter into the growth phase? The equal number of primary spermatocytes are produced only. Okay, one more important point, these primary spermatocytes are also present in diploid state. Means 46 number of chromosomes are there in this. How many? 46 number diploid state it is. Okay, up to here this is the what is first and second growth phase. Now the primary spermatocytes are enter into the third phase, that is the multiplication phase. That is the multiplication. That is the multiplication phase. Okay, that is the multiplication phase. Okay, what happened in this multiplication phase? Here meiosis process is continue in this. See what happened in this meiosis process. Okay, important functions are there. See, the single primary spermatocyte, when the single primary spermatocyte it is enter, it is completes the meiosis 1. It is completes the meiosis 1. Okay, the meiosis 1 phase is also called as a reduction phase. It is also called as a reduction phase. Means here the chromosomes number will be becoming a half. Reduction of number of chromosomes is called reduction phase, that is the meiosis 1. Okay, in this process what happened, after completion of the meiosis 1, two daughter cells are produced. These two daughter cells are called here secondary spermatocytes. These are the secondary spermatocytes. Now the secondary spermatocytes number of chromosomes are haploid, means 23 number of chromosomes are there. Means number of chromosomes are here become half here. Reason, this phase is the reduction phase. Meiosis 1 phase is the reduction phase. That's why the number of chromosomes are becoming half. This is the primary spermatocyte. Again, the primary spermatocytes are again which are involved in the meiosis 2. Again which are continues to the meiosis 2 division. In this meiosis 2 division, again daughter cells are formed. These daughter cells of secondary spermatocytes are called spermatids. These are the spermatids. And one more important point is there. Meiosis 2 is not a reduction division. Only meiosis 1 is the reduction division. That's why meiosis 1 is occur within the primary spermatocytes. Here 46 number of chromosomes are there. After completion of the first reduction division, means meiosis 1 division, number of chromosomes are becomes half. See, these two secondary spermatocytes are the daughter cells to this primary spermatocyte. Means the primary spermatocyte is the mother cell. These two secondary spermatocytes are the daughter cells. It is clear. And in mother cell 46 number of chromosomes are there. In this daughter cells 23 number of chromosomes are there. Means number of chromosomes are becomes half. That process is the reduction division. Okay, so after completion of the meiosis 2, immediately within the secondary spermatocyte, how many number of chromosomes are there in the secondary spermatocyte? Its offspring, means its daughter cells are also having, okay, its daughter cells are also having what is 23 number of chromosomes. These are having 23 number of chromosomes here. Means these what is, there is no chance for decreasing the number of chromosomes. See, the spermatids are having 23, its mother cell also having 23. Means the meiosis 2 is not a reduction division. Okay, meiosis 2 is not a reduction division. So it is very important thing, right? After that, see, up to here it is the sorry, this phase is the maturation phase. So this phase is the maturation. This is the maturation phase. This is the maturation. Okay, this up to formation of spermatid is the maturation, right? After completion of the maturation, the fourth phase will begin, that is the differentiation phase. That is the differentiation. That is the differentiation phase. Okay, so the differentiation phase, it is begins with spermatids. Okay, I'll what is draw these spermatids in new slide. Okay, here it is the fourth phase, that is the differentiation phase. Differentiation. So differentiation is also called as a spermiogenesis. It is also called as a spermiogenesis. It is also called as a spermiogenesis. See, these are the spermatids. The spermatids are the end products of maturation phase. These are the spermatids. The spermatids are small and round in shape, and which are having what is haploid number of chromosomes. Means haploid means what, 23 number of chromosomes are there. It is one. And one more important point is there. I'll explain this point here. Generally, it is the spermatogonial mother cell. Here, what are 44, right? Right. 44 plus here. Okay. See, in spermatogonial mother cells, 44 plus XY and 44 plus xy, total 46 are there, that's why. And these are all are diploid, that's why 44 plus xy, these are there. And what is 44 plus xy, these are there. And next, when the primary spermatocytes are enter into the maturation phase, in the meiosis 1 process, in the reduction division, in these two X and Y chromosome, one allosome is enter into one daughter cell and another allosome is enter into another daughter cell. Okay, and after completion of the meiosis 2, so these X chromosome contain a mother cell. It is again the same X chromosome transferred to their daughter cells. Y chromosome also same. Means in the 100 percentage of, right, in the total 100 percentage of sperms, 50 percentage of sperms are having X chromosome, 50 percentage of sperms are having Y chromosome. Okay, Similarly, in this, it is, and these are having X chromosome, these two are having Y chromosome. Say in the these what is into the differentiation phase, these all spermatids means what is, for example, one primary spermatocyte is there, it is produce the four spermatids, means this is the 1:4 ratio is there. So all spermatids are enter into the Sertoli cells, these are enter into the Sertoli cells, and within the Sertoli cell the differentiation process complete. Now the spermatids are produced. Now the spermatids, are produced. Here in this total 100% of sperms, 50% of sperms are having X chromosome, 50% sperms are having Y chromosome. Now these are the spermatozoas. These are the spermatozoa, and each spermatozoa it is present in haploid, and 23 number of chromosomes are there. 23 number of chromosomes are there. Then this is up to the spermatogenesis process here. Okay, in part 3 we will discuss what is the structure of sperm, and semen contain a number of spermatozoa, and we'll discuss in the remaining information we'll discuss in the next part 3. Okay, right, thank you.